This is a film about our project Biocare Marine. What we're doing is exploring the sea for new molecules that could be used for healthcare applications. In the film we hope to show you some of the techniques that we use in the lab for our research. In so doing we hope to demonstrate a regional capacity for the creation of high-tech products sourced from the local environment. We also hope to show that by working together, local researchers and industry can prosper in a synergistic way that could ultimately benefit the community.
One exciting area of our research is using marine polymers to create a range of sponge-like materials called Cryo Gels.
Here's Ian in the lab weighing out agarose and fish gelatin to make a Cryo Gel. Cryo Gels are composed of ingredients, including water, that form conventional jelly-like substances called hydrogels at room temperature. Dessert jelly is one example, but others are used for medical purposes, such as in wound dressings. Cryogel mixture is placed into a bath filled with ethanol, held at -20 degrees C. When hydrogel ingredients are frozen before they've had a chance to set, ice crystals form within the mixture, while the other components remain unfrozen and set like a conventional gel. When the material is brought up to room temperature, the ice crystals melt, releasing the water. This leaves a highly porous sponge-like material, the pores being a remnant of the presence of the ice crystals. Here we see the structure of a dried cryogel magnified many times using a scanning electron microscope. Cryogels can be made in a variety of shapes and sizes, like this cylindrical one. The cryogels have excellent shape memory. That is, they can be squeezed and returned to their original shape. Cryogels can hold relatively large volumes of liquid within their pores. Confocal laser scanning microscopy reveals the 3D structure of the cryogel and its sponge-like nature. Cryogels have a potential for a number of very useful applications.
Our team has been creating cryogels using almost exclusively marine materials and assessing their potential for use as dermal tissue regeneration matrices. These would be used as a potentially life-saving measure to repair the skin of patients with full-thickness burns.
These materials are biocompatible and non-toxic and are well suited to the cryogelation process. We've been using seaweed extracts and fish gelatin from fishing industry waste to make our cryogels. Human skin cells can infiltrate and multiply within our cryogels. This means that they offer a good potential for replacing large areas of deeply damaged skin, such as is found with burn injuries. To assess the regenerative potential of our cryogels, we seed them with human skin cells grown in the lab. Here's Matt examining some cells under the microscope. Typically, we would incubate them for up to 28 days and at regular time intervals examine some of the samples to check the extent of cell proliferation and infiltration. We can view the materials and the cells within using a confocal microscope. Here we can see a cross-section of a cryogel that has been infiltrated with skin cells. A fluorescent dye highlights the overall structure of the cryogel, here shown in blue, while another dye binds to the DNA within cell nuclei, here shown in green. By highlighting the cells, we can quantify the extent of their infiltration within the cryogel. The arrow shows the direction of infiltration of the cells that were seeded onto the lower surface of the material. By superimposing a grid of known dimensions, we can quantify this process. We can also assess the health of the cells by labelling specific structural proteins using fluorescent antibodies. Here, a protein called alpha-smooth muscle actin is highlighted in green and the cell nuclei are shown in blue. In this image, a different protein called fibronectin is highlighted in green. Here, we can see a single fibroblast cell adhering to the pore walls within a cryogel. Viewing the scaffold using a scanning electron microscope reveals the pore structure in exquisite detail. This animation demonstrates one potential clinical application of our Cryo Gels.
Injuries to the skin which will not heal by themselves can be treated using dermal replacement materials. Biocare Marine is developing dermal replacement scaffolds made from marine polymers using a technique called cryogelation. A sample is taken from the healthy skin of a patient with a severe dermal injury. Skin cells are isolated from this sample and cultured in the lab. The cells are then seeded onto the cryogel scaffold. These cells continue to multiply and infiltrate the material. The scaffold is gradually remodelled by the activity of the cells. Their enzymes break down the scaffold and replace it with an extracellular matrix. The material is now starting to resemble real skin. The surgeon places the scaffold onto the wound bed. Finally, an aerosol spray of lab-cultured skin epithelial cells is applied to the scaffold. After a few weeks, the wound should be fully healed.
Sea creatures have to compete for space and resources in order to survive. Marine bacteria, like those from other environments, try to outcompete their neighbours using a number of tactics, including the use of what are essentially chemical weapons, some of which we know as antibiotics. Biocare Marine is looking for new antimicrobial agents that are naturally produced by marine organisms. This comes at a time when many disease-causing bacteria are developing resistance to the current generation of antibiotics.
Biocare Marine holds a stock of extracts from bacteria that were isolated from fish and shellfish. We have been testing these extracts for their ability to inhibit the growth of other bacteria, especially those that can cause disease in humans, such as MRSA and E. coli. To do this, we first spread a layer of a disease-causing bacterium onto an agar plate. We then dispense a drop of the test extract and incubate overnight. The bacteria will have grown as a lawn over the surface of the plate. We then look for a zone of inhibition, i.e. a clear circle, around the drop of test extract. If these are present, we know that the extract possesses inhibitory activity against that particular bacterium.
Nearly everything we've seen in our film can be sourced from here on the Sussex coast. We've seen, for example, how we've been constructing tissue regeneration matrices for the treatment of burns injuries, using seaweed extracts and byproducts from the fishing industry. The sea also offers a great potential as a source of new antibiotics and indeed many other drugs that we can use in the fight against disease. If, as a community, we're to utilise the great riches the sea has to offer, then surely it must be protected.